I'm introducing Gabriel Popescu today. He is a professor in electrical and computer engineering here at U of I, and he received both BS and MS degrees in physics from the University of Bucharest, as well as an MS and PhD in optics from the University of Central Florida. He then did his postdoctoral research with the G.R. Harrison Spectroscopy Laboratory at MIT, and he has directed the Quantitative Light Imaging Laboratory here at Beckman Institute since August of 2007. He also served as an associate editor of Optics Express and Biomedical Optics Express, editorial board member for Journal of Biomedical Optics, and scientific reports. He has also written a book, edited another book, and has published over 170 journal publications, 210 conference presentations, 38 patents, and has given 220 invited or keynote or plenary talks. He also founded Phi Optics Incorporated, a startup company that commercializes quantitative phase imaging technology. Finally, he is an OSA fellow and SPIE fellow. Thank you, Dr. Popescu, for coming to speak with the Miniature Brain Machinery program today, and we're excited to hear your talk.
Great, thank you very much for the introduction. It's great to be back. I've given a talk here a few years ago already, so I think it's time for kind of an update since I can't recognize that many names anymore from our trainees, but glad to see that the program is going actually very, very well. So I'm glad to be involved in a certain way with the program. So let me see if I can share my slides here. Is this visible?
We're not seeing it yet. Oh, there it goes.
Okay. Is it visible now?
Yeah, it's in the PowerPoint view, not the screen share, or I'm sorry, it's not in the slide share mode.
Let me try that now. And hopefully nothing will, okay. Is this
Perfect.
okay?
Yes.
Great. So my talk is about phase imaging with computational specificity, or PICS for short. And I'm going to explain what that means. I would like to first thank a lot of people that contributed to this, to the results in these slides. And those are on the left-hand side, this is our group and people that some of them already graduated, some current ones. And I'd like to point out that a lot of these results, actually, none of them would be possible without the collaborations that we have across many different departments. I see Martha here. So Martha has been my collaborator from the very beginning, actually, from 2007. And we've had a number of publications so far, and we hope to continue in the future. So all the people that are highlighted here are directly involved in what I'm going to show you today. The outline is something like this. For those of you who don't know much about phase imaging, which I don't expect you do, I'll assume you don't. I'm going to start with some background and motivation for what we do in our group. Then I'm going to introduce a couple of methods that we call SLIM and GLIM. And I'm going to try to explain the difference between them and why we need to still develop new methods. And then the second part is about this PICS, which is essentially a combination of label-free imaging and artificial intelligence. So let me start with a little bit of background, and most of my talks start with this slide because it kind of illustrates one of the problems that we're trying to address. And that is that while fluorescence microscopy, as you probably know, is the most commonly used microscopy technique out there, is not without limitations. And you see right here as a quick illustration that these cells are fluorescing, but they're expressing actually fluorescence in their mitochondria. They become unhappy after a while. So after continuous imaging with fluorescence, there are a couple of things that happen. One could be due to the illumination. So the UV excitation usually is harmful to the cells, limits the continuous imaging for a long time. There could be chemical toxicity as well due to the tags involved. And there is also photobleaching. It's not obvious in this movie, but photobleaching means that after a while, the fluorescence kind of quenches and essentially the molecules stop fluorescing, and therefore you have a limited window of investigation. So we think that there is potential for other methods of producing contrast for such transparent structures. And here's a very old movie now that we obtained in our lab many years ago with Martha, actually. One of our first papers has this movie in it. And basically what you're looking at is a map of a contrast of a portion of a neural network where there is no fluorescence involved. It's essentially based on the refractive index contrast that the structures in the cells exhibit kind of intrinsically. So for that reason, you have no phototoxicity. The illumination is actually white light, very low power. So you can do this for many days continuously. And there is no photobleaching, obviously. So the refractive index is always there. This contrast never disappears. And in addition, the fact that we are actually quantifying this map in the sense of kind of think of it of a delay map. So where you see a little bit more red in the field of view, like here, it means that the light is slightly delayed with respect to, let's say, the background, where it's deeper blue. So having this quantitatively, you can actually relate it to something that is biologically relevant, like density of the cell, and specifically dry mass density, what's in the cell that's not aqueous matter or water. So based on that, you can actually quantify how cells grow. We came up with ways to analyze how vesicles and organelles are transported around. You can use the same information from a kind of an optics point of view to solve this inverse problem, meaning a three-dimensional reconstruction of the structure of the cell, and so on. So I'm going to show you a couple of applications in the next slide. I'm showing you this book. Shannon already mentioned that I sometimes write some books, but this is not to advertise or sell books. If you're interested to learn more about quantitative phase imaging, I'm happy to send you this PDF of the book. I don't want to be too technical because I understand the audience here is diverse, but just maybe quickly, there are two main ways that allow us to extract this phase delay, to obtain these maps that I showed you earlier. And one is called phase shifting, and the other one is called off-axis. So both of them involve interferometry. If you just use a single beam of light, you are not able to get this quantitative map that I showed. It's only the interferometry, essentially comparing two beams of light, that allows us to get into these details to render these contrast maps from unlabeled cells. The difference between these two is that in one case, in this phase shifting method, you are controlling the delay between a reference field, so a field that didn't go through the cells, with the light that went through the cells and through an imaging system like a microscope. And you are acquiring 4 different intensity images, and you kind of combine them in a fairly simple way. And this is how you get this phase delay map out. The other one, in the off-axis case, the reference comes at an angle, and the difference is that now this delay or phase information is incorporated in the spatial domain. It's in these fringes that are a little bit hard to see in this image, but imagine these little stripes that you kind of see. They get bent whenever the refractive index is higher than the background in the image. So you use a slightly different method of processing based on Fourier transforms, but at the end of the day, you end up with a quantitative phase map that has the same meaning as the previous one. And you may wonder, why would I bother with the first method to acquire 4 intensity images, which obviously is slower? Why do I need to bother when I can do this in a single recording from a single intensity map? And the answer is that in the second case, it is faster, but actually you give away something. Like always, there is no free lunch. And what you give away is actually a smaller field of view or smaller resolution, whichever you get to pick what you give away. And actually the sensitivity also of these maps is not as good in the first case. So for that reason, we have both of these approaches running in our lab, depending on the applications, we get to choose which to use. So let me first talk about spatial light interference microscopy. This is a method developed at Beckman very soon after I joined the lab. It's based, I'm skipping over a lot of history that we did as, I started in this field as a postdoc before joining Urbana, and we learn from our mistakes, and SLIM is the result of understanding a couple of principles that make these images better. So one principle is that it's always better to combine the interfering fields, bring them as close together in space as possible. So what that means is that the noise that the two beams are kind of encountering or experiencing is very, very similar when they're very close physically. So for that reason, then you get a phase map like this that is very, very stable in time. So you can image over and over and over, as I said, over many days, for example. And that this phase relationship is actually is controlled and very robust. So that is one thing that SLIM provides. The second thing is that it's using white light. It's actually using the illumination of a regular phase contrast microscope. We didn't change that commercial microscope at all. And SLIM is implemented as a module that attaches to that microscope. So using white light has another advantage. Again, not getting too technical from an optical point of view, but white light compared to lasers, monochromatic light, gives you a very, very uniform background. If you ever played with the laser pointer and if you shine it at the wall, you're going to see that there is a kind of distribution of intensities that goes from maximum to minimum very, very fast. People call it speckles. It's a manifestation of the coherence of light, which is very, very high for lasers. It's not so high for white light. So because of that, all these random distribution of intensities actually is averaged out for white light, and that's why we get these beautiful images with a very, very uniform background. So I'll leave that these technicalities like that, and just to bring it back into the previous kind of background slide, we do require 4 intensity images. We have an element here that is active. It's a liquid crystal modulator that provides a control phase delay between the 2 interfering fields. And we acquire 4 intensity images, and then we render them, render this phase map that is both stable, as I said, and with a very, very uniform background. So you're looking obviously at the neuron. This is an early paper that we published a while back now. Another thing is that SLIM can be, as I said, can be implemented as a add-on module to an existing microscope, and the microscope looks like this. So as far as a biologist is concerned, the experience of using this microscope is as usual. So we preserve that experience. So imagine you have a module that attaches here and everything else is the same. Another thing is that you have all the accessories that come with a microscope. So, you know, sometimes you need a perfusion chamber, temperature control, humidity, and so on. So this comes with the original microscope. So everything is there. And very importantly, fluorescence comes on the same optical path as the commercial microscope. So you can switch between SLIM and fluorescence as you like. And this is one example. If it starts working, okay, it kind of works. So on the left-hand side, you're looking at the SLIM image of CHO cells. On the right-hand side, this is, there we go. These are GFP expressed in mitochondria for the same cell. So you see, we went back from left to right, from SLIM to fluorescence. So now you can kind of get a specificity clue from the fluorescence, so you know exactly what you're looking at. So for example, if you kind of squint and you look carefully here, you are going to notice that these mitochondria that appear in the fluorescence have a correspondence in this refractive index map right here. Otherwise, it's very difficult to tell what is what. So this is kind of central to the theme of my talk today with this chemical specificity that is basically lacking in this label-free modality. And I'm going to show you later how we have currently we have other options on how to achieve that chemical specificity while keeping the benefits of label-free. SLIM is very, very sensitive. And here's an example why that is. I mentioned to you the reasons why it's so sensitive is because, again, it's very, very stable and it's using white light. And white light sometimes is better than lasers, especially in imaging. So, for example, because of all these sensitivity benefits, you are looking now at single microtubules. You see these lines here. These are single microtubules that we can image directly using SLIM. And as you know, these are 24 nanometer tubes. So very, very thin, very transparent things, but are very clear. And if you do some averaging in the time domain, you can actually get a much better SNR with SLIM. Normally, traditionally, these microtubules are studied with fluorescence, and you can see here actually photobleaching manifesting itself. In just a couple of minutes of continuous imaging, the fluorescence goes away. This is what photobleaching is actually doing, how it shows up. Here's another illustration on what we can do with converting these delay maps, these phase maps, into actually dry mass densities. And again, the point is that these colors are actually meaningful. Where you see more red, it means that there is more stuff, more dry mass content in the cell compared to the background. So basically, we did this somewhat simple experiment where we looked at how a neural network grows as a function of their confluences. So what you expect is actually, as you have these neurons sparse versus very dense, the neurons will have to produce a lot more protein here to make connections to one another. While here, the connections are already kind of made, so they will produce a lot more material. So this is exactly what we found, that between the sparse culture versus the dense one, we have a growth rate that's about a factor of 7, 8 smaller in this case versus here. So this is not so surprising. It's not kind of new science, but it was we wanted to test that we can quantify this actually in a very precise way. Here's another application that I'm not going to go too deep in it. It's about pathology. I wanted to mention that these fine details in the structure to the nanoscale can be used to actually retrieve information from biopsies or tissue microarrays in this case. Information that doesn't exist in the regular H&E stain slides. And one of that piece of information is about collagen that doesn't stain very well with H&E. But in order to make SLIM useful for these kind of applications, that again, I'm not going to talk too much today about, we had to build essentially a wide whole slide scanner, which is an automatic kind of instrument that scans the slides and produces these phase images quantitatively. So as you know, the commercial whole slide scanners are doing the same thing for H&E, where it's a lot simpler from an optical point of view. Just get an intensity map of those pink and purple maps. Here, of course, we needed to get the 4 frames that I mentioned to you earlier. The processing goes in the back. The user doesn't even see it. So everything is kind of scanned in real time. You can scan now entire slides. I think it's the first ever whole slide scanner based on quantitative phase imaging. And we published a number of papers on pathology applications. So just to illustrate what you get in SLIM or in phase that doesn't exist in H&E is actually this collagen or, if you like, tumor microenvironment that shows with very high contrast in SLIM because the refractive index is actually high and doesn't stain very well in H&E. And collagen has a lot of applications that is relevant to many different problems, but more recently it's been shown that for actually tumor pathology, it could be used as an intrinsic marker for kind of classifying the aggressiveness of cancer. So the collagen fibers tend to align in a certain way with respect to the boundary of the tumor that we were able to quantify. And as I said, we published a number of papers on this. But this is all possible just because collagen shows up with the refractive index very high. It's a very good signal for in SLIM. Another thing you can do based on this phase data is simply to scan the sample through focus, and you are going to get a certain sectioning, depth sectioning. I hope this is this visible to everybody? These
Yes.
movies are working okay? Okay. So for example, we'll look at the neural network again. This is the cell body. You see the top of the cell, then the center. This is the nucleolus. And then you see later how actually the neurites become in focus. And this is just the raw data coming out of SLIM. We understood that in order to get the pure 3D structure of the cell, we needed to do some more work, which involves a physical model for this image kind of creation and to be able to invert that model. So this took us a little bit of doing. I wanted to connect with this pioneering work by Emil Wolf, who's a kind of an optics giant in our field. And he published the first paper of reconstructing 3D structures of transparent unlabeled objects like cells based on holography data. So probably you've heard of holography. It was a Nobel Prize in Physics in 1971. The holographic data was kind of available, not very high quality, but theoretically, Emil Wolf put this paper forward in '69, showing how to go from this holographic data to the 3D structure of the object. It's very similar to X-ray crystallography, actually, turns out. The physics, the math is very, very similar. But nothing really much happened to it just because, based on this theory, just because the experimental access to this phase information was not really available, was not robust enough, was not high quality enough. But and we couldn't apply that model directly to our SLIM, which now is able to produce reliable phase data. We couldn't apply it just because SLIM has two differences with respect to what Emil Wolf published. First of all, it's in an imaging geometry rather than a scattering geometry that Emil Wolf wrote. And the second important one is that SLIM is using white light versus the monochromatic light that the model back in '69 was proposed. So anyway, what I'm saying is we had to do some extra work to actually generalize that model to what we needed. And it took us a while, but actually we managed to do it. I'm not going to get into the details of what this formula means in detail, but basically it's a combination. The sectioning along the z-axis, so this is our object. Imagine the function of the object, let's say refractive index in 3D. The z-axis is actually blurred by a function. This is a system of our microscope's function, if you like, the SLIM characteristic, but it has two components. So the numerical aperture has to be high, and also the power spectrum, meaning the spectrum of the light, has to be broad. So this tomography only works because we have both of them. We have high numerical aperture, which is easy to do, and we also have white light, which is actually not so easy to do, but we figure out how to measure phase from this broad white light spectrum. And we had this intuition. We know that both of these are actually very important. We knew this all along, but initially mathematically it wasn't very obvious. So finally, we're very happy when we got this to work. So at the end of the day, you go from the data that you acquire just by scanning the cells through focus or your object through focus, and then you go through this processing and you end up with this three-dimensional distribution of the refractive index. So these colors have nothing to do with fluorescence. They are actually values of the refractive index. And you see, it starts to look like a confocal image, except this cell is not fixed. There is no fluorescence involved at all. And these are more illustrations, a couple of neurons in 3D and based on the same kind of idea. And it makes me very happy that we celebrated 50 years from this pioneering paper by actually generalizing this problem one more time to second harmonic generation. So this is a more complex imaging system where we're using the nonlinearity of the sample to go from a single photon to two, if you like. So you illuminate with an infrared photon, and then you end up with green light on the other side. So this is a coherent nonlinear effect, and I'll leave it at that. But before our work, there was no model that would connect scattering with this second harmonic generation. There was no model to invert, to produce this tomogram from the raw data. So it also makes me very proud because this started as a project in my microscopy class. So I usually give them a final project to the students, and I have a list of kind of, existing methods to review, something like that. But I always have a list of things that I don't know how to do very well. So I told them I have this list of methods that you can review and present. That will be perfectly fine. And then I have this couple of things that if you're interested, we can work together. If it works out, we can publish it. I was thinking maybe optics letters or a smaller journal. And these students said, oh, I'm interested to do this. It turns out that actually came out the theory in the end when we got it right. It was so kind of interesting that we collaborated with the group in Colorado State who did the measurements. They sent the data and we reconstructed this, and actually we published in the best journal of optics at the end of the day. It took us a couple of years, but anyway, so that was made me happy, first of all, because it was a student project, and then because we're kind of celebrating 50 years from Emil Wolf's paper, who, by the way, I had the I was fortunate to have him as a teacher back when I was a grad student. Anyway, so I'm going to skip over the math and so on. The sectioning is even better with second harmonic fields just because of that nonlinear interaction. But essentially, the idea is similar, that you go from raw data that looks like this, you have all these effects of out-of-focus light and scattering and so on. And after you apply the calculations, then you end up with the clean kind of nonlinear refractive index signature. I heard some noise. Does anyone have a question or something? Okay. You can stop me at any time if you like. Okay, so many good things about SLIM, but then I think we published, I don't know, 50, 60 papers with SLIM, so we're very proud of ourselves. But at some point, you always find the limit of this method, whatever method you develop. And in this case, it happens that Matthew Wheeler from Animal Science Department a few years ago came up with this challenge to us. He said, oh, I read about this label free that SLIM can do. We have a very good problem for you. We want to look at the health of embryos, and these are bovine embryos. And this is an application where we cannot use fluorescence because this will be implanted, and you want to have a healthy pregnancy and so on. So this is a perfect application for SLIM. How do you think it's going to look under SLIM? So I said, wow, it's going to look great. Yeah, bring it on. And then basically we got these images that I consider semi-garbage. And you see the contrast compared to the images I showed you earlier in single cells and so on is very poor. The reason is that these embryos are an order of magnitude thicker than a single cell, right? So also, I learned later that the bigger the animal is going to be, the more lipid droplets it has inside, which is it's a very strong scatterer, and for that reason, your contrast is even worse than, let's say, for a mouse embryo. Okay, so we're kind of very depressed for a while, and then we started to think why this contrast is so bad. And we understood it. Actually, it wasn't too hard to understand that, in fact, what our interfering beams in SLIM do is one of them goes through the sample undeviated, unscattered. And basically we interfere that with the light that's scattered in the sample. That's how the interference geometry in SLIM works. The problem is, once you're at the scattering, a very strong scatter like an embryo or, I don't know, a spheroid or something thick, that unscattered light disappears very quickly. It's actually attenuated according to a Lambert-Beer's law. It's an exponential attenuation. that essentially the more thickness you add, the less of that field is left over. So what happens at the end of the day, you interfere a very weak unscattered field that acts as a reference in SLIM with a very strong scatter field due to the single and multiple scattering coming from the sample. So when you interfere a very weak beam with a very strong one, the contrast of interference is very, very low. This is kind of a basic law of interferometry. You want to have those as equal in power as possible to produce very strong interference signal. So once we understood that, we said, well, how can we develop a microscope where these interference beams are always equal in power? Even though they get degraded by scattering, by multiple scattering, they will remain identical in power. So it turns out, once I understood that the answer was just in front of us. This microscopy modality that is called the differential interference contrast or Nomarsky microscope, DIC, some of you probably have been using it for a while. So in this modality, the interference beams are this. You have an image of your sample that interferes with the identical replica of itself, but slightly shifted along one direction. So these two are identical, including in power, carry identical information. And if you pass both of them through a very thick object like an embryo, they will be degraded, but they will be degraded at the same pace. They will remain equal in power. So we figured out that this must be our solution. So basically what we did, we built a module that's very similar to SLIM, hence the name that's close. We call it GLIM. The reason for this gradient in the title is that actually this generates a derivative along one direction of the phase, not the phase itself. So it's the gradient along one direction. But the idea is the same. We collect 4 intensity images, we combine them, and what we get after combination is actually the quantitative gradient of the phase. So you see this shadow, there is dark here and bright here. This is if you take the profile of this bead and you take a derivative along one axis. This is the quantitative gradient. This can be integrated, the gradient, with some assumptions. It's not perfect. And then you end up with a quantitative phase, the same as I showed you with SLIM. So, okay, leaving all these technicalities aside, at the end of the day, this is the improvement. So here's where we were with SLIM, and this is what you can get with GLIM now. So this is just because we understood this very basic fact that you cannot interfere a very weak signal with a very strong one and expect high contrast of interference. It just doesn't work. So in this case, the two powers of the two beams are matched and actually interfere with high contrast. And you see the lipid droplets that I mentioned to you earlier. These are 1 micron to 2 micron diameter. Lots of them for an animal that's going to become a cow. Much fewer in a mouse, for example. So anyway, the improvement in contrast is actually very strong. So this is a clear example where you're happy with the method. We were for a few years with SLIM. So we did a lot of interesting things, we thought, and we published a lot of papers. And then at some point you have to realize that nothing is perfect. And in this particular case, the limitation was very clear cut. The moment you added multiple scattering objects, SLIM started to fail. The contrast was actually not great. So anyway, from GLIM now you can render these kind of interesting movies or you know, volumes of the cells inside and so on. So we are applying this to studying viability in embryos. Oops, I want to show this movie that I think is interesting. Come on. There. So this is actually a brain slice from Martha's lab. And this is actually a GLIM that was, we published another paper where we put the GLIM in reflection. So it may sound like a small difference, but actually what this allows us to do is to look at bulk tissues, including, I don't know, looking at your finger, for example. So obviously you cannot do that in transmission. We created a new geometry. And here, this is a portion of a hippocampus, as you can tell. And normally we wouldn't be able to see anything in here with SLIM, for example. It would be such low contrast. But here we actually see individual cells, obviously, sometimes subcellular structures, sometimes some connections between the cells. And based on this, actually our students, Martha's group and ours, worked together to put a SLIM system on top of an electrophysiology system from Martha's lab, and we're in the process of publishing this latest work with this combination, which I think is actually interesting. So this GLIM idea allowed us to do experiments that really we couldn't before. So let me go back to the original problem. So fluorescence has limitations, no questions about it. Photobleaching, phototoxicity, and so on. I provided this as a solution that I made it sound like it's a great, perfect solution, but actually it's not. And the main limitation with any label-free imaging is actually lack of specificity that I've already kind of alluded to. So this is the actual reason why we tried very hard to make our microscopes or the modules interface seamlessly with fluorescence, because we knew this up front, that if I really want to be specific to what I'm looking at, I have to be able to go back from fluorescence to SLIM or GLIM or whatever. So we knew this all along, that we can, we will give away specificity, and gain this non-destructiveness and kind of lack of photo bleaching and so on. But what happens in the past couple of years is that there is another solution to this problem that allows us to preserve the label-free imaging in the fact that it's non-destructive and gain the chemical specificity without much other loss or other compromise. And this is using advances in deep learning and artificial intelligence. And I don't want to sound like we've done this for the first time. We didn't actually. I was one that I kind of resisted all this trend with AI. I thought that too many people are doing it, cannot be good. But after a while, somebody twisted my arm and took our data and showed me what AI can do for us. And since then I was sold. So there is a number of groups, including Google, Allen Institute, and in our biophotonics group, the OSCAN group at UCLA, that really drove the field to new heights. And we basically borrowed what was out there, we adjusted, we advanced whatever we needed to do with the computational part to advance our use of our label-free data. So the first application was on actually doing sperm selection for in vitro fertilization, also a collaboration with Matthew Wheeler. So the problem was really, I don't know, trivial looking back. We were using SLIM to image thousands of sperm cells, like in one afternoon, in a few hours. And then it took a couple of undergrads 3 months to manually annotate, I don't know, the head of the cell and the midpiece, the neck and the tail, such that we can compute various dry mass contents and kind of do certain ratios and matrix and correlate them with success of in vitro fertilization. So this was a terrible situation. Our imaging was very fast. The analysis was slowing us down by months. And this is when Nahil Saab, a kind of computational expert, basically forced me. He said, can you give me that data with the manual annotation? And he came back a few days later and he showed me that actually the AI could do all that, what the students were doing for 3 months, could do it instantaneously. Once you train on a few hundred images, ground truth manual annotation, then the deep learning tool, the neural network, will be able to replicate that in no time and almost perfectly. So yeah, I don't have a lot more information on this. This paper has everything you need to know. But basically, this was so encouraging and kind of game-changing for us that then, of course, we started to wonder what else we can do with this approach. And we are now full force on AI. I think every student is doing something with AI nowadays, with deep learning. So the next was And this is what we now dub phase imaging with computational specificity. The next question was to go to more complicated cells, like your regular eukaryotic cells, and essentially infer or produce synthetic fluorescence images. So you take the label-free data from SLIM or GLIM, then you stain, then you image the fluorescence. In this case, let's say DNA or the nucleus. Okay, and then create these pairs of images, which is very easy for us to do because, as I said earlier, the fluorescence and the SLIM or GLIM are co-located. So it's very easy to create hundreds of these pairs and use it for ground truth, for training. And then once the neural network is trained, which can take anywhere from 1 hour to 10 hours, then you're done with fluorescence. The computer is actually simulating that fluorescence in almost with perfect accuracy. And not only that, one last thing is that then you can go to these compartments like the nucleus, for example, and then create binary maps and then compute dry mass, but very specific to that only nucleus, for example, or just cytoplasm, or I don't know, something else. So there is a kind of a double benefit for doing this. So the acquisition, as I said, is actually very easy. So this is a GLIM, happens to be GLIM, it could be SLIM as well. So automatically in a few seconds, you get, I don't know, a few hundred cells, very easy. Then you turn on the fluorescence, this DAPI. And again, in a few seconds, you got all those cells now in DAPI. And with simple thresholding and so on, you generate these pairs with hundreds of cells in essentially no time. And then you feed it to the computer. And then what you get out is actually this inference where, so here we use 2 channels, so DAPI and then DII, which is this cytoplasm dye. And this is the inference. And this is the neural network that we use. It's a UNet initially. Now we got a little bit more elaborate. We work a lot with Mark Anastasio now, whose lab is just, that's their expertise. So this is kind of the idea. We took an extra step and actually made this implementation of the, if you like, the synthetic fluorescence and put it in real time in the acquisition software. So basically, this is now the computational fluorescence in blue, and it's so fast, it takes about 60 milliseconds to infer it in this field of view. It's actually faster than the acquisition of SLIM. So that's why when you drag the stage from left to right, actually the computation keeps up, you see? So if I didn't tell you that this was fake fluorescence, you wouldn't know. So the experience is really as usual, except there is no fluorescence, it's all computation. So this can add a layer of benefits that can be applied to so many different applications. So at the end of the day, you create a movie like this. These are cells that have never been tagged with anything. They haven't seen stain in their lives ever. And to make it a little bit more interesting, we mixed 2 different kinds of cells, one metastatic and one non-metastatic breast cancer cells. And what you see is that actually, because we trained on both cells, the inference works in real time. Notice this is a 6-day movie that or a whole week that you wouldn't be able to do with fluorescence. You couldn't do fluorescence images for a whole week, imaging for a whole week without damaging the cells or basically killing them very fast. So in blue, you see DNA, in cytoplasm, cytoplasm is in red, and this actually can be done indefinitely. So you got the specificity from computation and the non-destructiveness from the label-free. So from here, the Q in QPI, in quantitative phase imaging, as I mentioned already, you can go back, create these maps, digital maps, and then get just the nuclear dry mass or the cytoplasm dry mass, and then you can add them together. You can do all kinds of interesting things. For example, we found that even though the metastatic cell is much smaller, so the area is much smaller, you see it very different in area. Actually, their dry mass is the same, dry mass density. So I don't know what it means, but it's kind of interesting. That the size is very different, but their concentration is identical. Something more recent was about using this label-free imaging with computation, with deep learning, to actually classify viable cells versus injured cells or dead cells. And this came out of a motivation that this came out of a visit that we did at one of the big pharma companies that they were demoing one of our instruments. And in the process of, discussing with them there, I learned that the way they test the viability of their cells from the bioreactors, which they have to do occasionally because their bioreactor costs about $250,000. So if the cells die in that bioreactor, that's $250,000. So they have to check the viability of the cells. And the way they do it, essentially they pull some cells in addition, then they add trypan blue. This is looks like a ink, blue ink. And it's a test that's so trivial and coarse that I couldn't believe it. These are multi-billion-dollar companies that all use the same thing. So basically, if this ink penetrates the cell, gets into the nucleus, it's a sign that the membrane is actually destroyed and the cell is not viable. But they told us, we have to do this so fast because in 10, 15 minutes, this dye is going to kill all the cells. So think about that for a second. You're testing the viability of the cells with something that kills all the cells in like 10 to 15 minutes. So I thought that was the craziest thing I've ever heard. And then we started to think how we could do this in a better way, just, getting essentially these morphology details of the cell with the label-free technique. And then to make it kind of precise, bring a computational tool that will be able to recognize, kind of associate the morphology with the viability. So that's exactly what we did. For ground truth, we took a kit. We bought a kit, a fluorescence kit that actually has two colors. So these are viable cells in blue. When the cells die, it becomes green. Okay, so this was our ground truth. We basically fed, again, very easy for us to acquire these pairs of SLIM or GLIM and fluorescence, fed it into the neural network. And at the end of the day, we have this. This is the ground truth. This is our PICS inference. And the accuracy is actually pretty good. And here we prove that actually for the labeled cells, their variability is actually going down much faster, which is not so surprising. It's very clear that any form of labeling will affect the viability of the cell. So anyway, now imagine you can do these real-time kind of movies. So this is label-free on the left. These cells were never labeled. And you see how they change color. They go from blue, healthy, to an intermediate. This cyan is an intermediate kind of injured state. And then they become green, which is a sign of dead cells. So this is actually, in principle, could be a solution for many different cell types. If need be, you can retrain this for your particular cell, for example, if the transfer learning doesn't work. But that takes, let's say, one afternoon or one night. You leave it run overnight, and then the next day, you have a label-free viability test. You'll never have to use any trypan blue, any stupid test like that, and it will always work. Of course, we got more ambitious after this, and we're working towards doing this in actually within spheroids and 3D kind of constructs that are a lot more interesting and challenging to work with. But I think this will have a lot of other implications, for example, testing drugs, as you know, creating spheroids or microtumor models in vitro, exposing them to various treatments. Efficacy of those treatments kind of translate into viability, right? Or a death assay, as they call it. So having a way to do this precisely without labels that interfere with your viability, I think it will be very useful. I wanted to show you one last thing, which is using that sensitivity that's very remarkable, I think, in SLIM to detect single viruses. And of course, it's SARS-CoV-2 kind of relevant, but in principle, any other viruses will work. So this first paper that we published last year was to prove that if you pair SLIM with, again, deep learning, you can not only just detect individual viruses that, as you know, are sub-diffraction limit. So SARS-CoV-2 is somewhere, I don't know, 60, 80 nanometers. But actually, you can classify different kinds of viruses, which was really surprising to us that it's possible to do from this label-free channel. So we mixed 4 different kinds of viruses. And Of course, SARS-CoV-2 is one of them, but also influenza, Zika, and adenovirus. And long story short, I'm cutting it very short here, but all the details are here. The confusion matrix looks like this. So combining 4 different viruses, you get 84% accuracy in selecting the SARS-CoV-2. Influenza A, almost perfect. The worst is actually Zika virus, 63%. We don't know exactly why that is. It's the smoothest virus. It has a hexagonal structure as revealed by SEM, and they tend to pack in a kind of very smooth fashion. So maybe that maybe lacks some signatures for us. Anyway, to be honest, this was more to kind of show our muscles, because I don't think there is anyone as unlucky to get infected by 4 different viruses at the same time. Probably our money or our main interest would be somewhere here. So to distinguish between SARS-CoV-2 and influenza, and it's already with 4 classes, it's working really, really good. If you just make SARS-CoV-2 versus influenza, it will be above 95%. Now, this is a test that in principle could be done in less than a second. So the moment you have viruses on a glass slide and you put it under SLIM, it takes 65 milliseconds for the deep learning to identify and classify the viruses. What we had in mind before, I mean, once this looked like it's working, there's a lot of potential here. What we had in mind was to actually create a breath test, meaning no nose swab or anything like, or saliva even. Just breathe on a glass slide, just put it under SLIM and let the computation do the rest. So over the past few months, we've been testing this actually in the clinic. So in Chicago, we have 60 patients with several slides each. Essentially, these are glass slides that come on one of those shields that are common in the clinic, and the patient just breathes normally, not much interference with the normal life. And then we collect those slides and we image them and we test them. So we're in the middle of validating this clinically, and hopefully we'll be able to go to FDA approval hopefully soon. This was part of an emergency grant supplement that we received from NIBIB. So with this, I'd like to stop here. And in summary, I talked about SLIM, that is very, very sensitive and is almost ideal for thin samples. So pathology slides, few micron thick, single cells, neural networks, you name it, including viruses. So very good technique for thin stuff. GLIM extends these benefits of label-free phase imaging to thicker samples. We have a lot more data that I didn't have room to include here, but this allows us to go to embryos, organoids, even animal models. We looked at like whole C. elegans, for example, brain slices, which I think is a very, very interesting system for us, very kind of complicated to image, but I think very rewarding. So we hope to continue this with Martha. And finally, this idea of using computation to take back that chemical specificity that we essentially give away, I think is going to open up a lot of different opportunities for us. We have a number of projects undergoing, probably about a dozen, where we use this similar approach to have label-free data as input and then output something that's chemically specific. Could be, I don't know, mitochondria, could be various kind of vesicles. Once you figured how to do that, then you can watch the transport over many days with no interference, with no loss of viability. And as I mentioned, I think viability will find different applications, especially if we generalize it to 3D structures. So I'll stop right here. I need to acknowledge that I have financial interests in PhiOptics that's commercializing both SLIM and GLIM. And with this, I'd like to thank you very much. And I'm open to questions. All right, thank you.
Well, while people are thinking up their questions, Gabby, I haven't heard you talking too long. You got all these new things you can do. So great.
Sure.
Yeah, the
Really
virus
amazing,
stuff
Gabby.
is really interesting.
Yeah, amazing.
What's
Gabby, I
the
have a question.
resolution in a cell? I mean, how, like, we know that redox state changes naturally in cells. And the question is it changing only in the mitochondria, or are there other cytoplasmic areas? Could that be resolved?
I don't know is the quick answer, but I'm definitely interested to try. In fact, we have an ongoing project precisely on mitochondria, and it looks like it's working in terms of the technical kind of, which is the first step, right? To show that we can identify mitochondria with label free. So that is kind of working. I mean, it is working, but we would like to add some, interesting add to that to add an interesting application when we're thinking to compare these metastatic cells versus non-metastatic. But I think something with redox, provided you help us with the ground truth to tell us what would be the ground truth using fluorescence, for example. Then we'll be happy to go and see if we can replicate that with label free, and then that would mean that we could do it indefinitely over long periods of time, many cell cycle. Who knows? I think this may have. June, I think you would be interesting with something with senescence. I think that is also kind of would be really cool. We'll see.
Yeah, yep.
So yeah, we're happy to try anything you think might be interesting.
Great. I see hands up.
So Gabby, the white light imaging that you showed the mitochondria, not mitochondria, but microtubules, right?
Yes.
So have you tried smaller structures like actin, filamentous actin?
Yes and no. So single strands of actin, no. But we did image years back. We looked at cells and how they change once you treat them with cyto D. So we basically destroyed the active structures, and the effect is very obvious, very brutal. So there was, I would say, almost not interesting. It was so clear. Yeah. There's no question about it. But now, if you're asking about single polymer strands of actin, never try that. So can they even be isolated? Can you isolate them on a piece of glass, or you're thinking inside the cell?
No, inside the cell, inside the presynaptic terminal of neuron junction. So it's a small space, and it might be like 2 or 3 layers of maybe not actin, not a single filamentous actin, but bundles of actin that you kind of see in TEM, but no other microscopy that I know of.
Yeah.
So let's say maybe the whole thickness may be a micron or 2 microns, and then it has 2 or 3 layers of network. We don't know, nobody knows, but that's in.
I would say the thinner the better, first of all. So it's not going to be trivial for sure. Even microtubules inside the living cell is a totally different ballgame compared to what I showed you. These were single microtubules on a piece of glass. These are from Paul Selvin from his assay that they're using. Inside the cell, the problem is much more complicated because you have all the background, right? Above and below, you have all these other inhomogeneities. So it makes it a lot trickier to identify single microtubules. Actin looks like, like cotton candy inside the living cell. So very complicated. To see single strands might be too much to ask for.
Do you do like confocal? Is it possible to have layers of imaging?
Yes, we have actually, I didn't have time to talk about it, something very new. We have a GLIM system on top of a confocal microscope. And it's so amazing and surprising to me that you can actually get GLIM images, and then with computation, you simulate the confocal. We call it artificial confocal microscopy. That was reviewed by Nature Photonics just recently. So yes, so we can have now confocal ground truth on the same field of view with this label free.
Mhm.
And I think a lot of, exciting opportunities there too. So
I see. So it has
Yeah.
the same step height or the Z stacking height as the confocal, or is it something different?
So natively, our imaging is not as good as confocal in terms of Z section. First of all, it's in the transmission geometry, you get coarser Z resolution just by the physics of it.
Mhm.
Confocal is so good in Z section because you're doing it in backscattering.
Mhm.
But what's amazing is that you feed these lower resolution images in, and the AI is spitting out confocal resolution
Mhm.
from a transmission input, which is amazing.
Mhm.
So basically, we can have label-free imaging with the same resolution as confocal, is what
Mhm.
I'm-
Okay.
and chemical specificity that learn from the confocal.
Yeah. Yeah.
So a lot of things that could be done, I think.
Right. Right. That's what I'm thinking. That with the AI, maybe this stack of filamentous actin network inside the cell could be resolved.
Well, what we need to do for that, I would say, is we need to team up with Paul Selvin and use his super resolution
Mhm.
to create the ground truth. You
Okay,
see?
I think I'm sorry to interrupt, but we do have another question from June, and then maybe we could see if there are any other people who have questions and then come back to this.
Yeah, I'll try to make it short because it's already after 5 o'clock. So you showed the image of the collagen matrix. Was it slim or slim? I think it was a slim.
Slim.
Yeah. So have you tried to kind of identify like or distinguish collagen from other like ECM matrix? For example, like pathological tissue has lots of the fibrin clot. So it will be actually very nice to distinguish collagen from this fibrin clot without any labeling.
No. So quick answer, no.
Yeah, but if you actually, I thought that the chemical specificity from your like
Yeah.
AI may actually kind of be worthwhile to think.
Yes.
Right.
So
Yeah.
as long as we have a ground truth for these 2 or 3 classes that you're
Yeah.
thinking, I think it's worth trying, and I'm already very optimistic. What
Okay.
we did in the meantime on collagen
Yeah.
is that now we have AI that specifically identifies collagen in no time. It's basically instantaneous from a pathologist.
Okay.
So I think to
Alright.
have 2 classes, I don't know, elastin or fibrin or something else is doable. It definitely
Okay.
works. Yeah.
Okay, thanks.